I'm really excited to be talking about a cancer that I'm very interested in studying, and that's medullary thyroid carcinoma. And what I'd really like to talk about today is how we are trying to understand medullary thyroid carcinoma in a bit more detail than we've been able to in the past. So my name is Priya Dedhia, and I'm an endocrine surgeon at Ohio State University. So I see patients and operate on patients with thyroid cancers, including medullary thyroid carcinoma. And I also have a lab where we study medullary thyroid carcinoma. So first of all, I just want to go over the talk that I'm going to give. And so first, we'll talk a little bit about what makes medullary thyroid carcinoma unique and how it's different from the other thyroid cancers. We'll spend a bit of time focusing on what the thyroid normally does, and then what the cells that start medullary thyroid carcinoma, what they do. Then we'll dive into talking a little bit about some of the research models that have been used to study medullary thyroid carcinoma and how we're trying to make progress with some new research models in order to understand tumor complexity a little bit better. And what we'll talk about are some of these newer techniques, which one of these techniques is single cell genomics. So I'll explain a little bit about that and what makes it so powerful. Then I'll talk about organoid or mini organ research models, which also give us another way to understand medullary thyroid carcinoma and the tumor complexity that's there. And then the last thing we'll talk a little bit about is the metastasis-on-a-chip model, which allows us to understand what cells can metastasize or spread to other organs versus what cells in the tumor might not metastasize or spread to other organs. So first, let's take a step back for a moment and let's think about what we think the thyroid does. So when most people think about the thyroid, they don't really think about the cells that cause medullary thyroid carcinoma, but rather they think about the general thyroid. So what happens with the general thyroid is that the anterior pituitary, which is a part of the brain, releases a hormone that stimulates the thyroid. And that causes the thyroid, most of the cells in the thyroid, to make thyroid hormones. And those are the T3 and T4 hormones. Those hormones then can act back on the brain and say, okay, hey, body, we have too much thyroid hormone. Let's stop this stimulation of thyroid hormone. So we have kind of a loop here where the thyroid hormones can act back and forth in order to control different aspects of the body function. So now let's talk about what these thyroid hormones do. So the thyroid stimulating hormone, it stimulates production of the thyroid, but it also can stimulate growth of these more common cells in the thyroid. And when your thyroid stimulating hormone is low, that leads to increased production of thyroid hormone and decreased thyroid cell growth. So now when the thyroid, these most common cells in the thyroid are stimulated, they release T3 and T4 hormones. These are known as the thyroid hormones together, and there are the two different forms because one of these forms is the active form and then the other is the storage form. Both of these together act on metabolism in the body. And so that's why you may hear of hypothyroidism, where the body feels a little bit slow, you can feel cold, you can have hair loss, you can have dry skin, weight gain, etc. Those are all things that are related to the thyroid's production of these thyroid hormones. But this isn't really what we're talking about when we talk about medullary thyroid carcinoma. So what is medullary thyroid carcinoma? Where does it come from? And how does it relate to these other cells that produce that produce the thyroid hormones? So what I have here is a picture of the thyroid, a cartoon picture of the thyroid. And what you can see is in these circles, these are thyroid follicles. So all of these purple cells are producing thyroid hormone as well as colloid, which is a protein that it gets secreted into the middle of this follicle. So these are the cells that release that thyroid hormone and then produce colloid. But those aren't the cells that give rise to medullary thyroid carcinoma. Instead, we have these little cells that are kind of in the corners and in between the thyroid follicles. And those are called the parafollicular cells or the C cells. And those are the cells that give rise to medullary thyroid carcinoma. So we'll take a few more moments to talk more about these parafollicular cells. So here is a picture of the actual thyroid, a normal thyroid. And what you can see are these big follicles again. And so the follicles have colloid in the middle. So all of this pink stuff that you see in the middle, in the what we call the lumen of these follicles, that's all colloid, and that makes it very clear to us that we're looking at the thyroid, and that the thyroid is producing these hormones as well as colloid. So the cells that are lining these follicles, you can see in the purple dashed line here, those are the follicular cells, and those are again those cells that produce thyroid hormone. So when we classically think about the thyroid, really what people are thinking about are the follicular cells of the thyroid, not so much the parafollicular cells of the thyroid. On the other hand, when we kind of look for these parafollicular cells, you can see them again in the corner in between the follicles, and you can see that they're a little bit different. They look different by eye from those follicular cells. So instead of having kind of more of an elongated or oval type of shape, these parafollicular cells have a circular shape, and they also have little dots in them. If you look really closely, you can see these little dots in them, and that's because they are secreting hormones in a certain way that allows them to pile up this hormone, which you can see as these little dots. So what are the differences then between these parafollicular cells and follicular cells, and why do we even care? So as I mentioned, parafollicular cells are the cells that give rise to medullary thyroid carcinoma, and follicular cells are the cells that we classically think about as thyroid cells, where they're producing the thyroid hormone. So first of all, the parafollicular cells, the hormone that they're really known for producing is calcitonin. In addition to calcitonin, they also produce a tiny amount of serotonin and another hormone called somatostatin. On the other hand, follicular cells produce the thyroid hormones, as I mentioned, those T3 and T4 hormones. The function of the parafollicular cells, it's mainly to regulate calcium, and I'll get into that in just a moment. On the other hand, as I had mentioned, the follicular cells regulate metabolism by producing those T3 and T4 hormones. So now, taking a closer look at what calcitonin does. So calcitonin is produced by those parafollicular cells in the thyroid, and those parafollicular cells release calcitonin when there is too high of a blood calcium level. That calcitonin can then stimulate the bones as well as the kidneys and the intestine to do different things that can reduce the calcium. So the calcitonin can stimulate the bones to take up more calcium in the bones so that there's less calcium in the blood. Similarly, it acts on the kidneys to reduce how much calcium the kidneys keep, and that means leading to an increase in the amount of calcium in the urine. So calcium is then lost in the urine. In addition to that, calcitonin can act on the intestine to prevent or stop the intestines from taking up additional calcium. And all of those things together can then lead to a lower calcium level in the blood. So when there's a high calcium level, if it's from eating or something else, then the calcitonin can help bring that calcium level back to normal. Or the calcitonin can bring that calcium level back to normal. Now, in terms of the way the parafollicular cells and follicular cells are developing, they come from different sources. So the parafollicular cells are neuroendocrine cells, so they have different mechanisms of how they're regulated compared to follicular cells, which are thought of as endocrine cells. Also, the other really important thing is that parafollicular cells are pretty uncommon compared to follicular cells. So you saw in that picture how you only had a couple of parafollicular cells compared to about 50 or so follicular cells. So 2% of 2% of the thyroid is parafollicular cells. So we'll look at why this matters. What does it matter that the parafollicular cells are so uncommon compared to the other cells? So when we think about thyroid cancer, most people think about follicular cell-related thyroid cancers. And so that includes differentiated thyroid cancer, like papillary thyroid cancer or follicular thyroid cancer. There are also other types of differentiated thyroid cancer. And in addition to that, there's also undifferentiated thyroid cancers, but all of these cancers come from follicular thyroid cells. The reason that that's important is because all of those cancers together make up the majority of thyroid cancers. On the other hand, cancers that come from the parafollicular cells, these are a much lower proportion of thyroid cancers. So about 5% or so of thyroid cancers come from parafollicular cells. And again, only 2% of thyroid cells are parafollicular cells. So what does that mean because this cancer is so infrequent compared to other cancers? Well, really what that has led to is that a lot more research focuses on follicular thyroid cancers instead of parafollicular thyroid cancers. And we really understand a lot less about parafollicular thyroid cancers and even the normal development of parafollicular or C cells. So whereas we know very clearly where follicular thyroid cells come from, there's actually been debate until just about 10 to 15 years ago in terms of where parafollicular cells even come from. So initially, it was thought that these come from neural crest tissue, whereas now, more recently, just in the past 15 years, we've realized that it actually doesn't come from neural tissue, but comes from endoderm, actually more similar to follicular thyroid cells. So now getting back to talking about medullary thyroid carcinoma. So as I mentioned, medullary thyroid carcinoma represents about 5% of all thyroid cancers, but it has worse prognosis than most of the differentiated thyroid carcinomas. And that's probably because we don't understand medullary thyroid carcinoma quite as well as we understand some of the follicular thyroid cancers. So medullary thyroid carcinoma is a cancer of those thyroid C cells or follicular cells. And there are two types that we think about when we think of medullary thyroid carcinoma, and that's hereditary or sporadic. And I'm going to talk about both of these in a bit more detail. All right, I'm going to then continue on into the genetics of medullary thyroid carcinoma. So medullary thyroid carcinoma, as I mentioned, has a sporadic component, and that actually makes up the bulk of medullary thyroid carcinomas. So 75% of medullary thyroid carcinomas are caused by sporadic mutations, which means that they're not inherited. And those mutations are mostly RET and RAS mutations, but there are other mutations as well. On the other hand, hereditary medullary thyroid carcinoma makes up 25% of all medullary thyroid carcinomas, and those are predominantly RET mutations. And when we talk about RET mutations, that can be in the form of multiple endocrine neoplasia, or it can be a non-MEN inherited medullary thyroid carcinoma. And again, both of those can be are usually RET mutations that are inherited or passed down from family member to family member. So why do we even care about RET and RAS? Why are we kind of thinking about these when we think about medullary thyroid carcinoma? So RET is a protein that's found on the outside of the cell, or what we call the cell membrane, and that protein can get activated and lead to signals inside the cell. So when that happens, when RET becomes activated, it leads to activation of the RAS pathway, which is a protein pathway that goes inside the cell into the nucleus of the cell. And that activates a whole bunch of other proteins, which then turns on, and then in addition to activating the RAS pathway, it can also activate the PI3 kinase and AKT pathway. All of those things can activate these proteins that really eventually lead to the cell turning on expression of genes that promote survival and growth. So why is that important? When that normally happens, it can lead to cell division. So for example, if there's injury to an organ, then you would want the cells to reproduce so that they can then lead to recovery of that organ. But when that happens in a dysregulated fashion, when it's not intended, that can lead to overgrowth of the cells and lead to cancer. So when we have activation of the RET or RAS pathways, then that can lead in an uncontrolled fashion, then that can lead to medullary thyroid carcinoma. So we know which RET and RAS mutations are associated with medullary thyroid carcinoma. So here you can see that there are tons of RET mutations that are associated with medullary thyroid carcinoma. And we've really taken advantage of this. So we've used different ways of studying these mutations and understanding what these mutations do, and we've been able to take advantage of this. So we have drugs that are able to target RET and RAS activation. So what you can see here is that same pathway I showed you, where we can see that RET can become activated. It can lead to activation of the RAS pathway as well as PI3 kinase, which then leads to survival and growth. And we can counteract this in two different ways. So some of the older ways that we've counteracted this is using a category of drugs called tyrosine kinase inhibitors, and those can block that RAS MAP kinase pathway to decrease survival and growth. In addition to that, more recently, we've identified a way to block that overactivation of RET, and that's with a drug called selpercatinib. There are other drugs that do that now as well, and those are all called RET inhibitors. The problem, though, is that even though these are pretty good drugs, the drugs are really only used for progressive disease. So once people develop metastasis, then we use these drugs. We don't really use them very often before developing metastasis because they can have side effects. But in addition to that, what we really worry about is that the tumors can develop resistance to these drugs. So we really hesitate to start these drugs until we know the tumor is progressing. So the problem is that we don't have a way of stopping the metastasis before it starts. And that's kind of what I'm part of what I'm interested in. And then the other thing is that we don't really have a way to treat these cancers once they've developed resistance to some of these drugs. So why don't we have more treatments for medullary thyroid carcinoma? Why don't we have ways to stop metastasis? I think part of the problem is the type of models that we've been using for medullary thyroid carcinoma. So you can see a picture of medullary thyroid carcinoma here on the side. You can see that all of these follicular or those circular looking cells with the little dots in them that are those secretory granules. And then you can see that these cells stain positive for calcitonin, which is that protein that they release in order to control the calcium levels in the body. Now, what models do we have to study medullary thyroid carcinoma? We have mouse models. That's been one of the big sources of studying medullary thyroid carcinoma. We also have cell lines, but we really only have a few of these cell lines. And cell lines are tumors that were taken from patients that are grown in a dish. But what ends up happening with cell lines is that they end up looking like just one cell. And I'll talk more about that in just a little bit. The other way that we can study medullary thyroid carcinoma is looking directly at the tumor characteristics. So we can take the tumor out and look at different aspects of the tumor to understand more about medullary thyroid carcinoma. But unfortunately, there are some problems with all of these models. And so I'll talk a little bit about that before we dive into the new types of models. So some of the problems are that mice, not surprisingly, they're not people. And so there are differences between the mouse and a human. And one of the differences that's especially important for medullary thyroid carcinoma is mice actually have many more C cells or parafollicular cells than humans do. So it's hard for us to know when we see something that happens in a mouse, is that actually going to be relevant to a human or not? And we can actually see that very clearly because there are some mutations that cause medullary thyroid carcinoma in mice, but we just don't see those in people at all. So we think there might be some discrepancies between the mouse models and what we see in people. The other thing is that, as I was alluding to, cell lines they don't convey tumor complexity because they end up looking like one cell instead of many, many cells. So what do I mean by tumor complexity? So when we look at a tumor, most tumors have many, many, many different cell types. So you might have one cell type, like the red cell type, which might promote metastasis. You might have the green cell type, which might promote inflammation. You might have the purple cell type, which can be resistant to dying or resistant to treatment with drugs. You might have the blue cell type, which can make new blood vessels form so that the tumor can spread more. All of these different cell types have different functions. And what that means is sometimes it can be hard to figure out which type of cell we have to treat. So one thing that we're kind of interested in is to figure out the cell types that can spread and lead to metastasis. So what are some ways that we've used to understand medullary thyroid carcinoma? As I mentioned, we can take the actual tissue from cancers when patients are having surgeries and try to look at different aspects of the tumor. So what has been done in the past is looking at the actual DNA, which is how we've identified those RET and RAS mutations as well as other mutations. We can look at the RNA, or what is expressed by the different genes. And then we can look at the protein as well, which is how we know that medullary thyroid carcinoma expresses calcitonin, and that it leads to activation of all of the downstream proteins uh, um, that have been activated by RET and or RAS that then can lead to genes that lead to proliferation and survival. So we've looked at all of these things, and we've definitely had benefits from that. That's how we've identified those inhibitors of the RAS pathway, and that's also how we identified an inhibitor of the RET pathway as well that we currently use in treatment. But that's kind of where things have stopped. And so what we're really interested in doing is looking in more detail to understand the complexity, because so far people have looked at the tumor as one whole tumor, as if it's one cell type. So what we really want to do is to understand it in a more complex system. So how can we do that? Some of the ways that we're interested in doing that is looking at single cell genomics, so understanding what each individual cell in the tumor is doing. And then we want to use organoid models, which, unlike those cell lines where they really they all look like one cell, organoid models look like many different kinds of cells. And then the last thing is we want to use this metastasis-on-a-chip model, where we can really see the types of cells that metastasize and compare them to the cells that don't metastasize. So I'm going to go into all of these models in a little bit more detail. Before I get into the details of these models, all right, well, I'll continue on then. So now, what has been done in the past is to use these tissues. So we take the tumor and then we do bulk analysis on them. So you can see all of these different cell types, the green, the yellow, blue, and the maroon colored cell type. And what we do is just put them all together in a tube and then look at everything together. So we can look at the RNA, and what we can see is that, oh, we have all of these different genes that are expressed. So the blue gene is expressed at these low levels, but we can't really tell, well, what gene is related to the green cell or what gene is related to the blue cell. So that's a little bit hidden for us. On the other hand, when we use this technique called single cell analysis, what happens is we can keep all of those cells separated, and then we look at the expression of the genes in each of those different populations. So instead of thinking, oh, this blue gene is expressed at really low levels in our tumor, what we might actually find out is, hey, look, this green population of cells expresses that gene at very, very high levels. So maybe we should target that gene. We should find a drug that affects that gene, as opposed to when we did our initial analysis with bulk RNA, where we couldn't really tell. So the reason that single cell analysis can be really important is because we can look at many different populations in the tumor, and then we can look at changes in tumor populations as well. So we can compare the cells before and after drug treatment. We can also look at our primary tumor and compare that to metastasis, so we can start understanding what is important for drug resistance and what is important for metastasis. So here are some of the data that we've developed in looking at medullary thyroid carcinoma. So what you can see right off of the bat, so the normal NAT refers to normal adjacent thyroid tissue. And what you can see just by looking at all of this globally is that there's a population in the normal thyroid that seems to go away in the medullary thyroid carcinomas. So that's that kind of lightish green population that you can see here. Also, you can see that there are many populations that dominate in the medullary thyroid carcinoma. So that's that salmon colored population, and then the yellowish, the orange and yellowish green populations. Those are now dominating in the primary medullary thyroid carcinoma. And what's really interesting is that we don't see all of those populations in the metastatic medullary thyroid carcinoma. We just see a couple of them, and it's really that salmon population that seems to be the most important population in the metastatic medullary thyroid carcinoma. So this is just a different way to look at the same information. So you can see that in most of the cancers, you have this very large component of that salmon population, and especially in the metastatic medullary thyroid carcinoma, which are these last few samples here, they're mostly all salmon colored cells, except for this one cancer right here, which has a different cell type. So now you can begin to see, we can begin to imagine, well, the questions that we want to ask are, well, what does that salmon colored cell do? And how is it different from all of the other cell types? And what does all of this mean? So the way that we're trying to understand that now, we can categorize all of these different cell types and look at the different pathways that these cell types are expressing. So the salmon colored cell type is right here, and the pathway that it's expressing most highly are these different pathways that can be upregulated or important in other types of cancers. So, for example, it's known that TGF-beta can promote cancers, and also Notch can promote cancers as well. So, of course, now that we have this information, that begs the question, if we target TGF-beta or if we target Notch, will that affect our ability to prevent metastasis or our ability to respond to drugs? So now that we have that information, well, how do we study that? Because the single cell information, it doesn't really allow us to test that in a system. It's just information that's given to us. So how can we study this in a system? I already mentioned that mice aren't quite like people, and I mentioned that those cell lines they only have one cell type. So how do we use a model that can let us understand this tumor complexity? So the models that we're really interested in are organoid models. So what are organoids? So these are mini organs, and the things that are important about these mini organ systems are that they're three-dimensional. So like our organs in our body, they're three-dimensional, which is different from a cell line, which grows on a plate and is therefore two-dimensional. The other important things about organoids is that they can grow for long periods of time. They have many cell types, so that's really important for our tumor complexity, that they're going to have many different cell types that are seen in the tumor. The other important thing is that they have a lot of aspects of that organ. So for what's relevant for medullary thyroid carcinoma is that organoids for medullary thyroid carcinoma produce calcitonin. So I'm going to show you that here in a moment. Similarly, if we make organoids for follicular thyroid cells or follicular thyroid cancers, they produce thyroid hormone as well. The other important thing about organoids is that they have some type of spatial organization. So you can see in this picture here, there are these little protrusions, or there's a circular formation where there's an inside and an outside, and there are areas that kind of are growing out. The other things that are important about organoids and why they can be a really nice system to study cancers, especially, and also medullary thyroid carcinoma, is that these organoids, they continue to have that complexity. So they have multiple different cell types. We can assess organ physiology. So we can look and see, hey, are we still producing calcitonin? Did we stop production of calcitonin if we treat with a certain drug? Also, compared to mice, they're pretty inexpensive to grow. So because they're inexpensive, we can actually treat organoids with many different types of drugs. So it allows us to do drug discovery and identify new drugs that we might not have thought might be relevant to a specific type of cancer. The other really important thing about organoids that's especially relevant for medullary thyroid carcinoma is that they can model human tissues, and they can be generated from rarer cancers or uncommon cancers and from biopsies as well. So how do we make these medullary thyroid carcinoma organoids? So what we do is when a patient has surgery, we take the surgical specimen, we take a portion of that, so we send part of it to the pathologist, and then we take part of it to the lab, and then we use enzymes to break up the tumor into individual cells, and then we plate all of these individual cells into different types of matrices or things that allow and support those tumor cells. And then we can add growth factors that are needed that your body would normally provide otherwise. And then we can grow them in a dish like this in little droplets that are three-dimensional. And then we can assess hormone production, we can look at gene expression, and we can assess responses to drugs as well. So we've started generating these medullary thyroid carcinoma organoids in our lab. We can grow them for long periods of time. So here's what they look like. And then they secrete calcitonin for long periods of time as well. So you can see that when we have other types of organoids, they don't produce calcitonin. But when we have these medullary thyroid carcinoma organoids, they produce pretty high levels of calcitonin in a long-term fashion. And then when we look at them under the microscope, we also see that they express markers of medullary thyroid carcinoma. So E-cadherin is a marker of epithelial cells, which medullary thyroid carcinoma normally expresses. And then we also see calcitonin. The other thing that I want to draw your attention to is that you can see differences here in the levels of calcitonin expression. So you can see some cells that have very low levels of calcitonin, other cells that have very high levels of calcitonin, and then there are some cells that have no calcitonin at all. So this suggests that we have that tumor complexity. We have cells that are different. They're all acting in kind of a different way. So we can test that then in future work. So now if we wanted to study this in more detail and figure out, well, which ones are the cells that metastasize or spread to other parts of the body versus which ones are cells that are the same. So for this, we work with engineers that have developed a model of a metastasis-on-a-chip. So I'm going to talk more about this metastasis-on-a-chip model. So what we've done here is we can take that tumor from a patient and grow that as an organoid, and then put that organoid in the metastasis-on-a-chip model and look and see what cells are going to go into a distant site organoid. So we have lung organoids, we can have liver organoids, and then we can just have empty matrix without any cells on them, and then look to see which cells will go to these different sites. And that will give us an idea of which cells might be more likely to metastasize or not. So this allows us to understand what populations might metastasize versus not. So that C1 population that I showed you from our single cell sequencing, we can now use this model to see, does that population actually metastasize? And then, more importantly, we can look to see the effects of drugs on metastasis. Are there certain drugs that are going to have a better effect on metastasis, even though it might not have the best effect on proliferation? So we've just started to develop this model here. So this is some of our early experiments in putting together this metastasis-on-a-chip model for medullary thyroid carcinoma. And what I'm showing you here in the gray, those are all lung. This is a lung organoid, and the green are medullary thyroid cancer cells. And then we've stained this lung organoid for calcitonin, and you can see these little dots of calcitonin, which all indicate cells that have traveled in from the medullary thyroid carcinoma into the lung organoid. So some of our future work then is going to be to take these cells back out and compare that to our primary tumor to see which cells metastasize versus which cells don't metastasize. So that is all of these things together then are gonna allow us to understand more about tumor complexity. So what you can see here is we might have a medullary thyroid carcinoma with all of these different cell types. You have a pink cell type, a green cell type, a purple cell type, a blue cell type. But it might be that only this pink cell type is the type of cell that can metastasize. And this cell can metastasize, get out of the primary tumor into the blood vessels, and then into the distant site. So our goal is to figure out what makes this pink cell type unique, and can we target that? And ideally, what we want that to lead to in the future is to see, and these things might be different from one person to the next. And so we want to see, well, if a patient expresses these patterns, then maybe this green drug might be better for them. And we can see that their organoids are more responsive to this green drug, then that might be a better treatment for that patient. On the other hand, it might be that a different patient, their organoids respond to this purple, this other type of drug. And so that might be a better treatment for them. So we can pick the unique types of drugs that are going to be most useful for that patient, and treat the patients with that specific type of drug. But there might even be the case where there are patients that need both of those drugs, where they have organoids that are going to not respond by either drug alone, but you really have to use both drugs. And we can then use both of those drugs together to treat those patients. So this is all, so a lot of this was work that was done in the lab, and a lot of this work was actually supported by ThyCa. So I want to, as part of, in conjunction with the American Thyroid Association, so I want to thank all of you for your contributions to ThyCa, and those are some of the things that we're working on in the lab. And really what we're looking forward to is having more participation from patients, so that we can get the tissues from patients and develop more of these organoid models and these metastasis-on-a-chip models, and understand medullary thyroid carcinoma much better than we do now. So I'd be happy to take any questions that you might have, or any discussion that you have as well.
So the question is, can medullary thyroid cancer with the increase in calcitonin cause migraines?
So definitely medullary thyroid carcinoma can cause migraines. And I think the other thing that's challenging about medullary thyroid carcinoma is we don't completely understand what calcitonin does. And so that's part of not having great models to really understand what calcitonin does. So I think even though there are sometimes things that we don't think is related to calcitonin, that might not actually be true, because we don't have great models to understand it. And so as I had mentioned in the talk, we only found out 15 years ago where parafollicular cells are coming from. So a lot of the things that we have thought to be true for, over 100 years, we're just recently finding out that's not actually true. So I think it is very possible that medullary thyroid carcinoma can cause migraines.
Hello, can you hear me? Okay. The second half of that question is, when the MTC is removed and the calcitonin is undetectable, might migraines stop?
Yeah, so I think that is further evidence that there's a high likelihood that that the calcitonin level could be associated with migraines. So I think that's very possible.
Yes, Can you tell me where the metastasis would be in the body with medullary thyroid cancer?
Yes, so it can go to a few different places in the body. It can go to the lungs, and the bones are common sites for medullary thyroid carcinoma, but it can also go to liver and other places in the body as well. First question from Wendy Hull: as you use these models to identify cell types that can be targeted, what kinds of drugs would you use? More of the existing drugs or something completely different? So I think we want to do both, actually. So the first question is, we actually don't know if selpercatinib or these tyrosine kinase inhibitors, how they affect metastasis specifically. So the first thing we're interested in doing is to understand if there is an effect on metastasis. And then the second part is to identify new drugs that might have a better effect on metastasis than the existing drugs. So especially the data that I had showed about the that one population might have a signature for Notch or TGF beta. We definitely have Notch inhibitors, so that's something that we're interested in looking into in more detail. Then another question is it true that Cytomel is not recommended for medullary thyroid carcinoma patients? That is true, and that's because of that unique nature of medullary thyroid carcinoma, where it doesn't fall into follicular thyroid cancers. So Cytomel is actually causing, basically what Cytomel does is it suppresses growth of thyroid follicular cells. On the other hand, we don't have something like that for medullary thyroid carcinoma, unfortunately. Okay, and then another question in the chat. Have you worked with tumors that do not secrete calcitonin? Let's see. There was a tumor that didn't stain positive for calcitonin. Do you have suggestions for additional biomarkers to monitor progression? Yes, so some of the things that we, so unfortunately, if calcitonin and CEA are negative, it can be pretty challenging to follow. Another thing that not our lab, but other labs are interested in are circulating tumor DNA. So that would definitely be a way in the future to follow for medullary thyroid carcinoma, because we might be able to detect RET DNA in your bloodstream in the future. If there is a RET mutation there, or even if there isn't a RET mutation, we may be able to identify specific components of DNA in the future. Another possibility is that sometimes chromogranin A can also be seen with medullary thyroid carcinoma, so that might be worthwhile assessing as well. In terms of donating tissue, so there are many resources that are available to donate tissue. I think you can, if you're interested in donating tissue, you can contact me directly, and there are companies that we work with that are nonprofit companies that really try to connect patients and just connect patients with researchers and try to minimize the barriers to carrying out research. So if you're interested, please email me, and then I'm happy to put you in touch with some of these companies. The next question is if studies are being done in the Netherlands as well. There are definitely studies that are being done in the Netherlands. Unfortunately, I don't know how to put you in touch with some of the researchers that are doing that work in the Netherlands. And then in terms of contributing DNA data worldwide, we definitely try to do our best to do that. So actually, all major funding agencies in the US require that you post the data so that it can be accessible to other people around the world. So we definitely do that. And then we also work with other researchers to try to combine data as much as possible. Thus far, it's been in the US. It has not been international, but I am very interested in doing that in the future. Again, if you want to email me, I can take a look and try to find out more information about who might be available in the Netherlands and who's doing this type of research closer to the Netherlands, if not directly there. Okay. Okay, so in terms of taking Cytomel. So Cytomel can be taken to instead of levothyroxine for symptoms, but it's not a treatment for medullary thyroid carcinoma. On the other hand, for follicular thyroid cancers, for differentiated follicular thyroid cancers, it's actually part of the treatment for some patients for follicular thyroid carcinoma. So for medullary thyroid cancer, if it may not be a treatment, but it can improve symptoms of hypothyroidism if levothyroxine alone is not working well. I hope that clarifies clarifies a little bit more about Cytomel. All right. So, okay. So this is a great question about undetectable mutation by Foundation One. So unfortunately, it is fairly common to have an undetectable mutation with sporadic tumors, especially. We actually don't know a large number of the mutations that are associated with medullary thyroid carcinoma. So that's one of the things that my lab is interested in doing as well. So in addition to doing that single cell genomics, I didn't show you some of the data, but we're also doing studies where we can look at all of the different mutations that might be associated with medullary thyroid carcinoma. And we've actually identified a couple more that have not yet been published. And so now we're kind of trying to do additional work to see if this is a real mutation that can cause medullary thyroid carcinoma, or if it might be a passenger mutation that's actually not associated with the cancer, but that's just seen in people as kind of a normal variation. That's a little bit of the challenge in finding mutations, is it's not always known which mutations are actually causing or related to the cancer versus which might be normal variations between you and me, for example. All right. Now, the next question is, are there any studies that you're aware of for MEN2B other than the natural history study at the NIH? I think so. We definitely include patients with MEN2B for our studies as well. So I think there are several places around the US that study MEN2B, but it might be different aspects of MEN2B. So we specifically focus on medullary thyroid carcinoma, but I know that there are other places around the US that might study different aspects of MEN2B. And then, of course, the NIH is kind of one of the forefronts in terms of leading research on MEN2B. But again, I'm happy to kind of give you more detailed information if you want to send me an email. And let me just share my screen here with my email address. Okay, so so there's my email address there. It's priya.dedhia@osumc.edu. So please feel free to email me and indicate that you had a question related to the ThyCa talk, and I'm happy to try to get whatever additional information I can for you. All right, I think those are all of the questions in the chat. Are there any other live questions?
I guess I just had one question about, as you gain understanding about how it's spread through the blood for disc metastases, I guess MTC strikes me as an uninformed person as a fairly sneaky cancer that sometimes isn't caught. There's no presentation until the patient is already like stage 4. So I think you were looking at ways to, if it's identified in a patient, ways to stop it from spreading. You had also talked about trying to pick up indicators of the RET in the blood, which seemed good, maybe a way to detect it. But I guess, are we going to learn anything that can be used to
The metastasis.
Well, I help identify the cancer in a patient before it spreads.
That's a really tough question. So we do know a little bit on how to identify medullary thyroid carcinoma before it metastasizes. The problem is that our healthcare system doesn't support testing of everybody for everything. So it's kind of dependent on how common a cancer is in terms of how often it's tested, how often patients are tested for that. So for example, colorectal cancer is very common. And so because of that, everyone gets screening colonoscopies. Similarly, breast cancer is very common in women. And so everyone gets mammograms. On the other hand, medullary thyroid carcinoma is quite uncommon. And so even though we can test calcitonin in everybody, we don't do that because I think it could bankrupt our healthcare. Like these types of things in general could bankrupt our healthcare system. Now, if there's a risk for it, then it makes a lot of sense to test for it. And for sure, when we know that patients might have a risk for it, we always test for calcitonin. But yeah, that's a pretty challenging question. And I think we're slowly moving towards being able to do a pan screen for multiple different types of cancers in patients, but we're not quite there yet. And hopefully one day we'll be able to get there though. And Hugo, I completely agree with you. You know, when I first started in doing medullary thyroid carcinoma research, I have to say I was shocked that it was in my training that people identified where C cells even came from and that we had it wrong for about 100 years, you know? So it's very, very surprising. And I think that's a big part of why we're a little bit behind in understanding medullary thyroid carcinoma. But I'm hopeful that with patients and supporters like you, and that we can all team together and make a little bit more progress in order to improve the way that people do with this cancer.